Hello. So today's topic for discussion is evolutionary foundations of human brain. This is a very vast topic that can't be discussed in one or two sessions. But I've tried to make it as short as possible, but also covering the important details which needs to be covered. So why evolution? Theodore Dobzhansky said that nothing in biology makes sense except in the light of evolution. So, in biology, if you want to understand the structure of an organism, why a certain behavior is observed in different organisms but not in other organisms, according to him, everything can be explained if we understand the evolution of all the organisms. It is partly true and might not be true in all the sense. Like if we consider that there's a gene which will code for a structure, that structure will be used for a certain function, and that function would dictate certain behaviors of any organism. This is not as simple as that. All the levels, this level of hierarchy is not followed in a linear sense. There's not a single gene for a single behavior. Everything comes down to a collection of structures or a collection of functions or a collections of genes, how they are expressed in different organisms. So it's not as simple as that. So we'll try to understand this in this upcoming in this section session. So I've divided this whole session into various episodes. We'll be considering the evolution as a story and try to binge watch it. Firstly, we'll try understanding only the relevant concepts of evolution. We'll not be covering all of the concepts of evolution in details because it's not the scope of this topic. We'll be considering all the relevant topics which are coming forward, going into the nervous system of Lamprey in detail. Lampreys are considered as the prototype or the model organisms for the early vertebrates which live on Earth around 500 million years ago. We need to get into the detail of the nervous system of Lamprey, and this would help us understand much. So we'll be covering nervous system of Lamprey in detail, which will help us understand a lot about human brain. Going forward into from Lamprey to early mammals, all the organisms which we think like fish, amphibians, reptiles, birds, which are considered as early vertebrates, only be covering the changes which happened from lamprey to mammals, not details of every nervous system. Going forward from early mammals to primates, and then finally, what are the major differences between the primates and humans? What makes us human? Concluding with a concluding with certain topics. So starting with the relevant concepts of evolution, the origin of a concept of evolution, concept of phylogenetic bush, and phylogenetic bush of vertebrates in detail. So, when we hear the word evolution, the first name that comes to our mind is Charles Darwin. While Charles Darwin gave the mechanism behind evolution, he didn't gave the idea of evolution. The idea of evolution came much before Darwin. In early 18th century, Carl Linnaeus, he divided organisms in different groups, which he called taxa. So he gave this taxonomy of life in his book called Systema Naturae in 1735. Linnaeus divided all the organisms which he seemed fit in different categories or different groups. Going forward in early 19th century, Lamarck gave a mechanism of evolution. He proposed a mechanism which was much different than Charles Darwin. It was nowhere similar to natural selection. He gave an idea of transmutation of species, like species which require a certain function converts into that function. There are no divergence, there are no new species are being formed. Just a single species is being converted into different species based on their requirement. But this idea didn't get popularized much. Around the same time in early 19th century, there was a debate going on between Geoffroy and Cuvier. Geoffroy stated, Geoffroy proposed that there's a common plan. There's only a single plan on which all the animals are built according to a single plan or archetype. But Cuvier debated that there are four different categories in which any organisms can be placed. So why was this debate? Cuvier, being a morphologist, he saw the morphology of organisms. So, seeing the phenotype of organisms, he could clearly identify there are four basic different phenotypes in which organisms can be divided. With a lot of evidence and recent studies, evolutionary development biology, the field of evo is going in favor of that there's a common plan and that all organisms are made or built according to a single basic plan. Then the evolution, the mechanism of evolution, the natural selection, it was not just given by Charles Darwin alone. Alfred Russel Wallace gave a very similar theory independently, and both Darwin and Wallace has a joint paper on the mechanism of evolution, the natural selection, and Darwin Wallace medal given for contributions in evolution. It was first given in 1st July 1908, marking 50 years of the contribution of Darwin and Wallace. So this was a little bit of history of how the idea of evolution and how the mechanism of evolution developed. But with the idea of Lamarck. Lamarck gave an idea that all the organisms are divided into groups, which are called taxa. He didn't scale them into different levels, but later on, some scientists and researchers made a scale, a phylogenetic scale, or scala natura, based on the systema natura of Linnaeus. They claimed that organisms are in a scale. Some organisms are lower in a scale, some organisms are higher in a scale, with God being at the top and inanimate objects being at the bottom. But this is not true. All the organisms are evolving, all the organisms are diverging into a different group rather than climbing on a ladder which gives them which makes them more superior or more advanced than any other organisms. Mammals are not more evolutionary advanced than fish. They are just adapting to what environmental constraints they are being put to. So terms like paleo, archaea, neo, etc. all are being discouraged. We can call organisms early or recent based when they diverge from their common ancestor, but they are not primitive, they are not old, they are not lower organisms than the ones which are divided more recently than them. So, from phylogenetic scale, the idea of phylogenetic bush came, which is more appropriate. There is a common ancestor, which is called the node, and then from the common ancestors, organisms are getting divided. This can also be represented in the form of a cladogram, which is traditionally made in a horizontal format, but in the future slides, further slide, I've made them in a vertical format for the ease of understanding. So as we can see here that fish, birds, reptiles, mammals, all are in the same plane. All came to the same level. Just the fish diverged much earlier than reptiles and birds or mammals in the evolutionary timeline. So considering the 600 million years of evolution, how human brain or how humans evolved from their common ancestors. The common ancestor which we'll be considering in much of the detail would be vertebrates. But vertebrates diverged from chordates around 520 million years ago, and there is a certain cephalochordate called Amphioxus still alive. They their nervous system and the vertebrate nervous system. Will touch upon the comparison of these nervous system in detail in last, but we'll be focusing mainly on the vertebrate nervous system. So vertebrates 500 million years ago diverged, branch diverged from them called jawless vertebrates. They didn't have a jaw, they are aquatic. So lamprey is considered as the prototype model organisms for jawless vertebrates, and jawless vertebrates are the earliest vertebrates which were present. Further going into gnathostomes, which are with jaws. Chondrichthyes are cartilaginous fish. Sharks are considered as a model organisms to understand the cartilaginous fish. And then from the aquatic lifestyle, the organisms evolved into terrestrial forms with paired limbs called the common ancestors are called tetrapods. All have paired limbs. 360 million years ago, a branch diverged, which are now amphibians, prototype organism frog, and amniotes. Amniotes are the organisms which lay eggs with an amniotic cavity. What advantage does an amniotic cavity gives to an organism? Amniotic cavity prevents the egg from desiccation in a terrestrial environment. They don't have to. Amphibians came to a terrestrial environment, but still they need an aquatic body to lay eggs so that their eggs don't dry up. But with the evolution of amniotic cavity, now the organisms could get far away from aquatic lifestyle and can lay eggs even on terrestrial environment without them drying up. In amniotes, now this is not just one branch diverging and the other lineage following into what we consider as mammals or a human lineage, mammalian lineage or the human lineage. Now there is a divergence. Great deal of divergence around 300 million years ago, pan reptilian and pan mammalian group diverged. In pan reptilian, then 170 million years ago, birds diverged from the paraphyletic group of reptiles. Pan mammalian 200 million years ago, monotremes diverged, which consist now of platypus. And 150 million years ago, placental mammal diverged from marsupials. Marsupials are pouched mammals which have pouch, example kangaroo, and placental mammals, which includes primates and humans. So as we can see, birds and reptiles have diverged a lot and a lot earlier than we consider, and they can't be considered as a prototype ancestor, or their nervous system can't be considered for comparison with a mammalian nervous system. See how much this is true in further slides. So this is an overall basic evolutionary lineage or the phylogenetic bush of vertebrates. Going forward into the details of the nervous system of the earliest vertebrate, the lamprey. Lampreys are aquatic organisms. They live in freshwater regions and they represent the most ancient lineages of vertebrates which are still alive today. The major brain regions of a lamprey brain can be seen in this figure. They have a telencephalon, a diencephalon, mesencephalon consisting of optic tectum, the hindbrain continuing into the spinal cord. So we can clearly, with the pineal gland, we can clearly see that all the major brain regions, all the basic structure and functional organization of a brain, which we considered as a mammalian brain, was present in these early vertebrates, which were present around 500 million years ago. So the basic parts of the brain consisting of a prosencephalon, which can be divided into a telencephalon, diencephalon, a mesencephalon consisting of midbrain, hindbrain, with consisting of pons and medulla. Everything is present in a lamprey's brain. This is a schematic diagram of lamprey brain showing different parts. And as we can see, these are color coded. Telencephalon having pallium, subpallium, and hypothalamus. Hypothalamus was earlier considered as a part of diencephalon, but recent evidence suggests that hypothalamus is a part of telencephalon. Diencephalon consisting of thalamus and related areas like prethalamus and pretectum, midbrain, and hindbrain. So all the parts are present in a Lamprey's brain, major regions. So, are only the major regions present, or the further details of the nervous system is also similar in the Lamprey brain? Let's look. This is telencephalon. Telencephalon में hypothalamus has a hypothalamo-hypophyseal neurosecretory system. So this proves that the endocrine system is well developed in a lamprey. Telencephalon has pallium, which is subdivided into two major divisions, which are lateral pallium and the medial pallium. Lateral pallium has regions for vision, somatosensory area, the motor area, and the olfactory regions. And this pallium has a three-layer like structure. Not a true three-layered cortex, but a three-layered like structure. Going into the details of the neural circuitry, the brain circuit, the olfactory bulb is connected to the lateral pallium. The lateral pallium is divided into an ipsilateral and contralateral, and both the parts, both the sides are connected. Does not have a proper well-defined corpus callosum, which we read about in the human brain, but still both the parts, both the areas, both the sides of the lateral pallium are interconnected. Lateral pallium is further connected to thalamus with the striatum, optic tectum. They have a well-developed retina with connections with mesencephalon, which is the optic tectum. Now we'll see the basal ganglia circuitry. If we see this basal ganglia circuit, this can easily be passed as a human brain basal ganglia circuit. This has all the pathways like a direct pathway, an indirect pathway, or a direct pathway. So not only the major structures like telencephalon, diencephalon, but the intricate details of the basal ganglia circuitry, including the D1 and D2 receptors in the striatum, everything was evolved or was developed in Lamprey's brain, which is considered as early vertebrate, which developed 500 million years ago. Not just these circuits like brain circuit or the basal ganglia circuit, the reinforcement learning circuit like lateral habinula, medial habinula, lateral habinula, which conveys information when there's an absence of an expected reward, not just a reward and punishment pathway, learning pathways. Even the absence of an expected reward can modulate the dopamine release in these organisms. The medial habinula conveying information for fear and reward. All these circuitries are present in the lamprey's brain, and the motor projections of the lateral pallium going into the optic tectum or the spinal cord, the reticulospinal, the MLRs, which are the mesencephalic locomotory regions, obviously connecting to the striatum, subthalamus, basal ganglia, the whole circuitry. The connections, not just the regions, but the connections are also very similar to a mammalian or even the human brain. In context to the motor connections, motor projections, the lateral pallium is being connected to spinal cord. In spinal cord, there are CPGs called central pattern generators, which are quite similar to the CPGs present in the spinal cord of a mammal. But the major difference is that the CPGs which are present in a lamprey have a very few patterns, if I may say. So they are only for locomotion or for feeding. Not a complex behavioral repertoire are present in lampreys which are present in mammals and human spinal cord. So diencephalon is divided into three parts: the pretectum, thalamus, and the prethalamic regions. The pretectum modulates the motor activity on visual input, closely related with optic tactum. They have a thalamic region, not a distinct thalamic nuclei, which are present in a human or a mammalian brain. But as we discussed, there is habinula divided into lateral and medial habinula and pineal organ present, and also prethalamus. Going caudally into midbrain, lampreys have a well-developed eye with a lens, retina, and also six extraocular muscles. So to innervate those six extraocular muscles, there is an oculomotor nucleus. There's a well-developed optic tectum getting information from the visual input. Other nuclei in brain consist of the MLRs, which we already discussed in context with the motor function, nucleus systemic, periventricular nucleus, which is considered as a homologue of periaqueductal gray. So, a number of nuclei are present in midbrain which are closely similar to the mammalian brain. Going caudally to hindbrain consists of pons and medulla. We saw that there's third nerve which is present, and as we can see, there's a fourth nerve which is present, which interestingly exits dorsolaterally, just like a mammalian brain. Not from sides, not from front, dorsolaterally, quite similar to the mammalian brain. Third and fourth nerve done. Then we have fifth, seventh, ninth, and tenth nerve present in a brachiomotor nuclei, which is considered as a homologue to SVE. The trigeminal nerve, the fifth nerve, which is present in lampreys 500 million years ago, was also divided into three branches: ophthalmic, maxillary, and mandibular. But they don't have a visceral motor nuclei, which gives branches to smooth muscles or glands. So autonomic system was not developed at that time. Eighth nerve, which is the auditory nerve, is called the octaval nerve in lampreys and is a part of octavo-lateral area. Lateral area is lateral line system, which is present in aquatic organisms to detect the vibrations or the electrical stimulations which are coming from water. As we study in a human spinal cord, the dorsal roots are sensory receiving inputs, and the ventral roots are motor, which are giving the outputs. Exactly like the Bell-Magendie law, these dorsal spinal sensory roots are present in the spinal cord of a lamprey and the ventral motor roots. So, with all these detailed information, we can clearly agree that the blueprint of a mammalian nervous system has been developed before in the lamprey, and we can agree with Sten Griller. He is the head of the circuits controlling action and the revolution, the Department of Neuroscience in Karolinski Institute, and the group have been doing extensive research in the lamprey's nervous system for past 2 decades, and we can't disagree with their conclusion when they say that the blueprint of nervous system was made 500 million years ago. So the question which comes to anyone's mind would be, then what changed in these 500 million years ago? Let's see. So we discussed in details about the nervous system of the lamprey. Going forward into the vertebrate phylogenetic bush comes next the cartilaginous fish, then amphibians. Will be discussing the divergence of the amniotes into birds, reptiles, mammals, and see only the changes which are happening in their nervous system. All the blueprint, the structures which we discussed in the lamprey's nervous system are present. Just something, if something is added or subtracted, we'll just discuss only those changes. Starting with the cartilaginous fish, the model organisms are sharks. So these are the schematic diagrams of brain of a lamprey, which we have already seen. This is a schematic diagram of a brain of a cartilaginous fish. We can see all the parts of a brain from telencephalon till hindbrain are present in both of these organisms. Going into the details of the cartilaginous fish, the telencephalon has enlarged and become more complex. The lateral pallium and the medial pallium were the only two parts of the pallium in the lamprey. Now the pallium has been divided into four distinct regions, namely the lateral medial, but also a dorsal and a ventral pallium. The subpallium is further divided into regions like striatum, pallidum, and also a preoptic region. Hypothalamus, which was just a group of neurosecretory cells in a lamprey, is now has five distinct regions. In diencephalon, which consists of thalamus and its related regions, now they have discovered some cells called as calretinin positive cells. These calretinin positive cells are present in a human in intralaminar nuclei. So the origin, if we say the evolutionary origin of intralaminar nuclei started in cartilaginous fish. They don't have a thalamus with distinct nuclei, but they have specific cells which are present in intralaminar nuclei. And the function of intralaminar nuclei is corticothalamic cortical connections, which are pretty newer connections, if I may say. They have a isthmic organizer as lampreys, but lampreys isthmic organizer didn't form a cerebellum. All the major parts are present in a lamprey's nervous system except cerebellum. So now in cartilaginous fish, this isthmic organizer, which is present in midbrain hindbrain boundary, starts forming cerebellum. The cerebellum is relatively large and divided into a spinocerebellum and a vestibulocerebellum. Only two parts. Going to tetrapods, amphibians, the prototype organism is frog. Keeping all the schematic brain diagrams of lamprey, cartilaginous fish, and a frog together, we can see clearly that all the major parts from diencephalon till hindbrain are present from lamprey to cartilaginous fish and even in frogs. We can see that there's a division of olfactory bulb into a main olfactory bulb and an accessory olfactory bulb. The optic tectum or the mesencephalon is well developed and enlarged, but cerebellum has become relatively smaller. Telencephalon has medial pallium, which is now large with multisensory inputs and has been considered as the homologous to hippocampus. Subpallium is now well developed. They have a distinct dorsal and ventral stritopallidal system. So now the pallidum is divided into a dorsal pallidum and a ventral pallidum. The dorsal pallidum is mainly connected with striatum for the motor functions, and the ventral pallidum is connected with nucleus ambiguus for emotional processing. Diencephalon, the pretectum is now divided into three parts. So specializations are happening. The regions were already present, but now they are divided into either distinct nuclei or two separate parts. Like the olfactory bulb is divided into a main and an accessory olfactory bulb for one system for the olfactory sensation and the second system for the pheromones. Separate vomeronasal system going to the accessory olfactory. Mesencephalon has now been enlarged as we saw, and pronounced expansion of optic tectum and torus semicircularis. The optic tectum is homologous to superior colliculus and torus semicircularis is homologous to inferior colliculus, making the quadrigeminae in the mesencephalon. In the hindbrain, the cerebellum is relatively small. There's not much evidence suggesting why it happened, but there are few theories, and one of the theories suggests that in cartilaginous fish or the sharks, which we consider as the model organisms, they had to manipulate or they had to move in a three-dimensional space in water compared to frogs, which had their locomotory constraints mainly in plane on the land. thus requiring less of the function of a cerebellum. But these are all theories. Spinal cord may, as the organisms developed limbs, paired limbs called tetrapods. So now they have distinct brachial and lumbar enlargements, which are present in a mammalian spinal cord. And they have three separate ascending pathways: a dorsal pathway, a ventrolateral pathway, and also one dorsolateral pathway. Going diverging the tetrapods into the amphibians and amniotes. Amniotes, there are no model organisms which can be considered that these are amniotes. Just these are a common ancestor of the all groups like birds, reptiles, and all the mammals. So there are some features which can be considered as common to amniotes, which are first the loss of a lateral line system. Lateral line system we discussed was for the aquatic lifestyle to sense electrical or vibrations coming from water. As these organisms have developed the amniotic cavity, amniotic membrane, now they don't have to lay their eggs in water, and they can live their independent life in terrestrial environment, don't require a lateral line system. Telencephalon for the first time is now divided into layers. These are true layers, not some collection of cells which would appear like layers. This is true three-layered cortex with principal cells, the pyramidal cells, and their dendrites connected to each other and extensively branched. The hypothalamus is now receiving inputs from limbic system and also regions associated with memory. In diencephalon, thalamus has been expanded and has highly differentiated structure. With the division of myomeres were group of muscles which are present in earlier vertebrates because they don't require much of a manipulation of the locomotory system in aquatic lifestyle. So now in the terrestrial lifestyle with a paired limbs, the myomeres are now divided into individual muscles. And to innervate those individual muscles, there's the subdivision of spinal cord motor columns into discrete, topographically arranged motor pools. These are some common features of amniotes had a three-layered cortex, a true three-layered cortex, and we know that mammals have a six-layered neocortex. It's called neocortex, but neo words like neo and archicortex should be discouraged. So we might call it a six-layered isocortex. So mammals have a six-layered isocortex. Reptiles preserved the three-layered cortex of amniotes and also developed a group of nuclei called a dorsal ventricular ridge. Interestingly, birds have this dorsal ventricular ridge but don't have a three-layered cortex. They have a group of nuclei called wulsts. So their pallium is not divided into layers. So they don't have a true cortex. So how did this divergence happen? We might say that the common ancestor of birds and reptiles had a three-layered cortex and also had dorsal ventricular ridge. And they diverged from amniotes and they developed a dorsal ventricular ridge. And then the group diverging into birds lost that three-layered cortex but preserve the dorsal ventricular ridge. That's the most logical explanation of why birds have a whole and a dorsal ventricular ridge, reptiles have a three-layered cortex and a dorsal ventricular ridge, and mammals have a six-layered isocortex. Clear from this slide that birds and reptiles have diverged a lot and can't be considered for comparison with the mammalian brain or mammalian nervous system. So if we are going into the evolution of human brain, we will follow the lineage of mammals and will not compare the behaviors of birds or reptiles or the structures of avian telencephalon for a mammalian nervous system. This is the same divergence, the amniotes diverging into panreptilia and a panmammalia group, and then further dividing into birds a paraphyletic group of reptiles and mammals. We'll be going into the lineage of mammals, but just touching upon the structures of avian and a reptilian telencephalon. So these are the primary functions which telencephalon considered for, like odor detection, spatial navigation, sensory processing, or sensory inputs, multimodal integration. And we can see that despite having very different structures, both reptilian and avian telencephalon can subserve these primary functions. Like reptilian telencephalon is divided into four areas, like a lateral pallium, medial pallium, dorsal, and a ventral pallium. Avian or birds don't have a three-layered cortex. They don't have a pallium, but they have a wulst. They have a dorsal ventricular ridge. Their dorsal ventricular ridge is divided into a ventral mesopallium, a nidopallium, or a arcopallium, not arcopallium. And these all functions can be subserved by different structures. Thus, this can be considered as an example of a convergent evolution. Convergent evolution is the evolution of similar function with different structures, meaning that they don't have a common ancestor which has the same structure for the same function, and they diverge in evolutionary tree. They have a phylogenetic bush relationship, and then they parallelly evolved the same function from a different structure. This was just in brief about the reptilian and avian telencephalon. Going back to the mammalian lineage, from a three-layered cortex, how did mammals evolve a six-layered isocortex? To evolve, they added three layers, which was due to the expansion of outer radial glia cells, which are forming layers in the embryological development. So, in comparison, the first layer remained the same and the superficial layers, but second layer is homologous to the fifth layer and third layer is homologous to the sixth layer. The primary efferents are coming into the superficial layers and the outputs or the efferents are going from the deeper layers, as was in the three-layered cortex, and similar connections are still observed in the six-layered cortex. Fifth layer give output to corticospinal outputs and sixth layer give output to thalamus called with corticothalamic outputs. The second and third layer have intertelencephalonic connections called corticocortical connections, which are important for association of different inputs coming to all the different cortices. In mammals, we saw that early mammals are the monotremes, which include us, then going to marsupials, and then finally placental mammals, which are primates, including humans. So all the mammals have a six-layered neocortex or the isocortex. So what changed? What evolved in the 200 million years ago from when the bonotremes diverged? The marsupials have more sensory areas. The sensory cortex is now divided into five specialized regions rather than two or three regions which were present in monotremes. So sensory specialization happened. This is the case in all the mammals, all the diverging mammals. All the sensory inputs have been increasing in mammals, and for accommodation of all the information which is now being given to the nervous system or the brain is evolving to accommodate all that sensory inputs and making sense or making meaning out of that sensory information. In placental mammals, for the first time, a motor and a premotor cortex has been developed. Initially, the functions of a motor cortex were done by sensory cortex itself. They were getting information and then stimulating or giving effector information to specific muscles for locomotion or any manipulation. This motor and premotor cortex and also corpus callosum. Corpus callosum helped integrate information coming to different hemispheres of the brain. So the mammals are diverging into a very vast variety from whales, which are aquatic mammals, to bats, which can fly, has wings, and n number of specialization. But all these specializations are just because of their environment. Whatever environmental constraints these mammals were getting, they were evolving different structure, different phenotype, different morphology to survive in that environment. And it would be very exhausting if we'll be covering each of these mammals in detail and how and why they developed a sense of echolocation, which is present in bats or even dolphins, and all the different specializations which they have been acquiring. So we'll be focusing mainly on what changed from early mammals to primates, and then finally going into a difference between the primates and humans and what exactly makes us human in the next part. So we have covered the relevant concepts of evolution, the details of the nervous system of lamprey, and why lamprey can be considered as a blueprint of the mammalian nervous system. Then we went from fish to early mammals. The lamprey was the blueprint, and cartilaginous fish, cerebellum was added. Amphibians got a well developed striatum with a distinct dorsal and ventral striatopalliidal system. Amniotes had a first time true three layered cortex. The birds, reptiles, and mammals diverged. Birds had wolfed and dorsal ventricular ridge. Reptiles preserved the three layered cortex and developed the dorsal ventricular ridge. Mammals have a six layered neocortex, and in mammals from monotremes to placental mammals, the regions or the specialization of the cortex increased the sensory specialization, and in placental mammals there was origin of premotor and motor cortex, and also corpus callosum. So for now, this is it. I'll be covering the remaining topics in the next part. Thank you for listening.